Thank you all for joining us today. We have the great pleasure of welcoming Dr. Anthony Komaroff as our speaker. He will be discussing what pathological changes may cause symptoms of long COVID. As a quick reminder, these sessions are recorded and the lectures will be uploaded to the University of Utah's Project ECHO website, as well as the Bateman Horne Center's website in the next 2 weeks. So a little bit on Dr. Komaroff. He is the Simcox Clifford Higbee Distinguished Professor of Medicine at Harvard Medical School and is a senior physician at Brigham and Women's Hospital. Dr. Komaroff has published over 270 research articles, book chapters, 2 books, and served as the editor of the autobiographies for 2 Nobel laureates on the Harvard Medical School faculty. In the past 20 years, his publications have been primarily focused on myalgic encephalomyelitis, chronic fatigue syndrome, also known as ME/CFS, and the biology and clinical consequences of infection with human herpes virus 6A and 6B. Dr. Komaroff has been a member of multiple committees convened by the U.S. Department of Health and Human Services, NIH, CDC, and National Academy of Sciences. In recognition of his achievements, Dr. Komaroff has been elected as a Fellow of the American Association for the Advancement of Science and the American College of Physicians. It is a true honor to have you here, Dr. Komaroff. I'm going to turn the time over to you.
So I'll begin. Thanks so much for inviting me to participate in this session. It's a topic that is very interesting biologically, but also because long COVID has become so important to society socioeconomically. It's a very important issue to understand. I'm going to cover 4 questions today. How the symptoms of long COVID are shared with other post-infectious fatigue syndromes, what some of the underlying pathological changes appear to be in long COVID, particularly the reactivation of latent viruses, as well as the COVID virus, SARS-CoV-2 itself. And then finally, to speculate, but with a lot of empirical evidence to support it, about how the underlying pathophysiology of this disease causes the symptoms of the disease. The symptoms of long COVID are very similar to those of ME/CFS, which I've studied for a long time, Dr. Bateman has. But what's less well known is that they're also shared with a whole bunch of well-documented post-infectious fatigue syndromes. That is, acute infections, very well documented, with a variety of bacteria, viruses, even fungi, and protozoans, that after the acute infection has been treated and has subsided, some people are left with a lingering chronic illness that's essentially identical to long COVID. So long COVID is, in my opinion, just one example of post-infectious fatigue syndromes. How common is long COVID? There are many studies of that. One of the best, in my opinion, recently published, is a study of over 30,000 PCR positive cases compared with controls, 60,000 plus controls who were never infected. All of those folks followed for 18 months with repeated online questionnaires. And among the PCR positive people, those only 52% had returned to full health after 18 months. 42% described themselves as partially recovered, but 6% as not recovered at all. The risk factors for non-recovery in that study were being hospitalized with acute COVID, older age, female sex, lower socioeconomic group, and a past history of several chronic illnesses. Now, what are the possible triggers, particularly viral triggers of long COVID? Well, there are quite a number, I would say 8 to 10 in the literature so far, all of which seem to be playing a role in some cases, not necessarily in all. The four I'll focus on today are persistent reservoirs of the SARS-CoV-2 virus that elicits an ongoing immune response, injury to many organs by the virus during the acute infection that then requires a chronic inflammatory response to try to repair the injury, even if the virus is now long gone. Third, the reactivation of neurotropic pathogens that most human beings have within us for virtually all of our adult lives. And those are primarily several herpes viruses, almost all of which are neurotropic, as well as capable of causing lifelong infection. And also there's a possibility that endogenous retroviruses in our genome may be contributing in some cases. And then last, there's evidence that infection with SARS-CoV-2 disrupts the gut microbiome in a way that leads to inflammation and autoimmunity. So before I mention the evidence in support of each of these, just a few quick words about viruses in general. Most viruses that we encounter multiply within us for a while, but then are fully eradicated by the immune response. Others, as I mentioned, the herpes virus is the best example, multiply for a while and then become latent in some of our cells for the rest of our lives. Of the ones that multiply for a while and get eradicated, some of those, even though the virus is eradicated, you can no longer replicate and produce more virus, it leaves its nucleic acids and antigens harbors within the body, and that can elicit an ongoing immune response. And for the viruses that become latent, they can reactivate. And when they reactivate and start multiplying, that elicits an immune response. And then finally, the endogenous human retroviruses. When the Human Genome Project completed its first pass at its work, one of the remarkable discoveries was that 8% of our genome is constituted with ancient retroviral sequences. And at first, people assumed that this was just the detritus of evolution, that these sequences were totally inactive. They couldn't make virus or do anything that affected our health. Well, there's still no good evidence that they can make full retroviruses, but there's growing evidence that their genes, some of their genes, can be activated to produce viral proteins, and that those viral proteins can then go on to elicit an immune response or affect human health in other ways. That's not the topic of our discussion today, so I'll say no more about it, but it is interesting and it may well be playing a role in some people with long COVID. So what's the evidence that the SARS-CoV-2 virus itself can remain in the body for a long time? Because when this COVID epidemic began, the assumption that most virologists, because coronaviruses tend to be this way, was that the immune response would probably fully eradicate the virus from the body within weeks, a few months. But now it's clear, and there's one study that was just published this last week. It hasn't even yet been peer-reviewed, but it's from the NIH. I'm sure it's going to be published and soon, and I think it's very important. They completed autopsies on 44 patients with acute COVID-19, including 4 who died not from acute COVID-19, but from something else entirely. They used really highly sensitive assays for both nucleic acid and protein and for replicating virus. And what they found was the viral RNA remained in tissues throughout the body in all of the patients for as long as 230 days after acute COVID-19. And all of those tissues included the brain and all areas of the brain. And this was true even when the nucleic acid and antigens could no longer be detected in the nasopharynx or in the blood. And it was even true in the people who had very mild acute COVID at the beginning. The virus, it was shown by this study to be replicating in organs other than the lungs for at least 7 days after the onset of acute disease. There are multiple studies shown at the bottom of this slide that support the persistence of viral RNA and antigen in people with long COVID. Months after the virus has left the nasopharynx, you can still find it in intestinal tissues, liver, and stool. The symptoms, GI symptoms, correlate with the presence of the virus, residual virus in the gut. The B cell response over time strongly suggests that there is a persistent reservoir of virus in the body. That, as I said earlier, the virus, the SARS-CoV-2, can alter the gut microbiome in a way that makes the gut more pro-inflammatory and can trigger autoimmunity. And then finally, the persistence of viral RNA and antigen is much more common in people with long COVID than in those who have recovered from COVID. And the evidence for that is a study published just in the last couple of weeks. Small study, 37 patients with long COVID compared with 26 patients who recovered from acute COVID, all of them PCR positive. And they all had repeated blood samples over 12 months. The strength of this study was that it used ultra-sensitive assays for the spike protein of SARS-CoV-2. And what they found was that very low levels of the viral protein are detectable chronically in 60% of the people with long COVID and in none of the people without long COVID. That is, they had COVID, but they fully recovered from COVID. So that's an impressive study. Small, will need to be repeated, but I think will prove to be right. There are various features that distinguish people with long COVID from those who fully recover from COVID or from those who never got COVID in the first place. Increased antibodies to SARS-CoV-2 antigens, increased antibodies to EBV antigens, Epstein-Barr virus, one of the herpes viruses, lower levels of cortisol, increased activation of B cells, decreased CD4 memory cells, exhausted T cells, and increased production of several cytokines by CD4 and CD8 cells. Putting it all together, persistent antigen from the virus, reactivation of latent herpes viruses, both of which elicit an immune response and lead to a chronic inflammation, appear to be central to the pathology of long COVID. I'm going to skip this slide and just ask you to accept the fact, in the interest of time, that there's a lot of evidence of reactivation of Epstein-Barr virus, one of the herpes viruses in this illness. Now I want to turn to how this underlying inflammatory state, possibly triggered in different people by different triggers, but how it leads to the symptoms of long COVID. And I'll ask you to begin by thinking about three questions. What do you feel? We've all been sick. We've all gotten the flu. What do we feel like when we're sick? Why do we feel that way? And what chemical signals trigger those symptoms? When I say feel sick, I'm talking about fatigue, difficulty thinking, feeling achy, headache, poor appetite, poor sex drive. People with long COVID or people with ME/CFS often say it's like having the flu, except that it never goes away. So these sickness symptoms are not only a universal human experience, but you can, if you think about it, you can tell when your pets are sick. They behave in ways that indicate that they're sick, ways that are resonant of the way you feel and behave when you're sick. And in fact, when people study this sort of, these sickness behavior symptoms are present in most of the animal kingdom. So they've been evolutionarily preserved, and possibly for a reason, possibly for this reason. When our symptoms cause us to be much less active physically, mentally, when we sleep a lot more, when we eat and digest a lot less, when we have less sex. As a result, we are utilizing a lot less energy, which preserves our ATP stores, our energy stores, to fight the infection. This sickness symptoms and the change in our behavior is a protective response that preserves our energy stores to help fight the infection. So when the infection has been eradicated, what should happen, what does happen when we get the flu and other transient time-limited infections, is the symptoms go away because they're no longer needed. As I said, it's seen in most animals, even in vertebrates. It's evolutionarily preserved. It's supposed to be temporary because the threat to us that we have to fight is usually temporary. But in people with post-infectious fatigue syndromes, that stimulus that triggers the sickness symptoms must persist. And we can speculate as to what is causing it to persist, but I've already suggested several. Retaining the viral RNA and antigen in the body, disrupting the gut microbiome, reactivating latent viruses, all of that could be that ongoing process that keeps the sickness symptoms going. The theory goes further to say that within the brain, in this picture in the hypothalamus, there is a group of neurons, a nucleus of neurons, whose sole function is to generate sickness symptoms. At times we're sick. That nucleus of neurons is activated whenever the brain's innate immune system is activated. And the brain's innate immune system is activated whenever there is inflammation in the brain on the left, such as if there's an infection in the brain, or whenever there's inflammation anywhere else in the body. In my mind, one of the most interesting discoveries in physiology over the last 20 years has been the fact that when there's inflammation anywhere in the body, the gut, the bladder, anywhere, that sends signals through two routes, through the blood and also up the vagus nerve to the brain, that activates the immune system of the brain. It's as if it's an early warning system where the gut is saying to the brain, I got trouble down here, you may have trouble coming, turn yourself on because you're going to need to deal with it. And that now is solidly established scientifically. The part of this theory that had no empirical corroboration was the existence of a sickness nucleus of neurons. However, in the last 2 months, but particularly in the last 2 years, 4 different groups, all studying rodents, not humans, have identified exactly such nuclei in the hypothalamus and in the brainstem. These nuclei could be triggered when a viral infection or any kind of infection, in this case viral, in the brain led to inflammation in the brain. They could be triggered when systemic inflammation sends signals to the brain that activates the brain's immune system. And as I said, they could be triggered by alterations of the microbiome that causes systemic inflammation. So this theory is attractive, not fully proven, but I think should be the basis of some future research efforts into treatments that can improve the symptoms of long COVID and other post-infectious fatigue syndromes. So in summary, these post-infectious fatigue syndromes, like long COVID, have underlying biological causes. They are not the amplification of anxious symptoms. They are not, except in rare instances, in my experience, people seeking secondary gain. I had COVID, now I can claim to be ill. I'm sure that happens. It happens with a lot of different illnesses, but very rarely. And there's now abundant evidence of altered underlying biology. An infectious trigger, immune activation, neuroinflammation, autoantibodies, dysautonomia, oxidative stress, defective energy metabolism, a hypometabolic state, pro-inflammatory gut microbiome. Didn't have time today to show you all of the evidence for that, but it's robust. And that these abnormalities that exist in people with long COVID, ME/CFS, and other post-infectious syndromes are all connected. Most of them are capable of reinforcing each other, which leads to the potential for ongoing potential vicious cycles where something that makes oxidative stress worse makes inflammation worse and leads to the generation of autoantibodies. They affect each other. They probably cause sickness symptoms by activating the brain's immune system, which then activates a group of neurons dedicated to generating those symptoms. That's what we know so far about long COVID. The good news is, you have probably heard from other speakers, is that there is a huge investment now in studying the underlying biology of people with long COVID. And I'm optimistic that whatever is learned about that will prove to be applicable to people with other post-infectious syndromes and will lead to improved treatments for all of them. That's it. Thanks very much.
Dr. Komaroff, that was great. I'm going to open the floor up to questions because I know that I personally have some, but I have heard many of Dr. Komaroff's lectures, and every time I do, I learn something new. So let's open the floor to you guys. What sort of questions do you have for our guest speaker today, as well as our mentor panel?
I have a question. I'll start with the hard one. So, Dr. Komaroff, how do you tie these concepts in with post-exertional malaise, as opposed to just fatigue?
Yeah. I'm not sure, Cindy. I think there's two possibilities that come to my mind. There's probably many more, including some that are true. One is that exertion further depletes energy stores in an organism whose energy stores already may be depleted and whose capacity to generate energy molecules may be deficient. There's also a very interesting recent paper that claims that human beings with experimental mental fatigue have a buildup in a particular part of the brain of one neurotransmitter, glutamate. And that appears to explain in a fairly persuasive set of experiments that there may be this fatigue, mental fatigue molecule building up in a particular part of the brain, and that could be connected to the phenomenon of post-exertional malaise. But I think what you'd have to do, and in fact, two groups are doing it, including a group at Columbia and at Cornell, are doing studies of the metabolism of people before they exercise, while they exercise, and then repeatedly in the days after they exercise, to see whether exercise has triggered metabolic changes that track with the symptoms of post-exertional malaise. That's where I would put my money, trying to understand the physiology of post-exertional malaise.
Dr. Brown?
Yeah, that was fascinating. I feel like I have mental fatigue molecule buildup some days, especially after the last 2+ years.
Yeah.
The stuff you posted or showed about the RNA or the protein identification, I think is really interesting. Do they know the intracellular location of those things? Because I think about all the pathways that are dedicated to, you know, clearing abnormal proteins, et cetera, and whether they're, you know, being expressed by TLRs and sort of repeatedly goosing the immune system. That's my first question. The other thing is I know you mentioned like B cell phenomenon in the spleen. Have they looked at marginal zone B cells or anything like that as far as their kind of interplay between immunoglobulin production and things like that?
Well, the study I referred to looked only at measurable spike protein in plasma.
Okay.
It didn't look at the effect of that spike protein on toll-like receptors or on the immune response. So, but it wouldn't surprise me at all if they're looking at that. I just don't know. The paper I was referring to did not address that. That's all I can say.
Yeah, that's really fascinating. The other thing we've seen is that a lot of our cardiopulmonary exercise tests for patients who have long COVID, showing a lot of mitochondrial dysfunction as well. Wondering if you have any thoughts kind of of the literature you reviewed, how that might be linked.
Yeah, there's a group at my hospital led by David Sistrom that has done invasive cardiopulmonary exercise testing, where they're sampling blood in the venous system, blood from the heart at the same time as peripheral blood. And they are finding mitochondrial abnormalities, energy metabolic abnormalities in people with long COVID that are identical to those they also find in people with ME/CFS. No one has done those studies, to my knowledge, in any other post-infectious fatigue syndromes, but I think it's only a matter of time before they will, because my bet is it's shared pathophysiology across all those syndromes. But there appear to be two deficits, as I understand their work. One is deficits in the ability to produce ATP, but also deficits in some cells in the ability to utilize ATP for whatever their metabolic functions are. So energy metabolism, the ability to make ATP from oxygen, glucose, fatty acids, or amino acids via the urea cycle, all four sources of energy generation, of ATP generation, appear to be compromised for sure in people with ME/CFS and preliminary evidence in people with long COVID. So something is wrong with energy metabolism in people who feel that they don't have enough energy.
And I think to one of the points that you made, Dr. Komarov, is that in peripheral tissues, there may be still viral remnants that may be triggering mitochondrial dysfunction in the first place. There's been some interesting studies out of Germany in particular with HHV6
Yes,
and mitochondrial dysfunction. So that's certainly a point of interest here moving forward.
good point, Dr. Yeoman.
And I'll just add that Dr. Prusty, I think is who Braden's referring to, I was just looking this up, has done, I've heard him give talks, and he feels like there are a number of viruses that when they reactivate can trigger local mitochondrial fragmentation in adjacent cells. And I think we could learn a lot more about that in this complex situation with COVID.
I very much agree. There's one other wrinkle that I didn't, for the lack of time, didn't want to raise. It's only been raised by one paper, but from very good people. And it suggests that SARS-CoV-2 itself can, in some people, integrate its genome into the human genome, just the way a retrovirus does. Well, how can it do that? Since it doesn't have reverse transcriptase, which the retroviruses use to integrate their genome into the human genome. And the argument is these endogenous retroviruses that we all have within our genome have genes that make reverse transcriptase. And they argue that that's what happened in the case of SARS-CoV-2, several cases that they studied, that this RNA virus, which should be unable to integrate into a DNA human genome, is nevertheless able to do that because of these endogenous retroviral reverse transcriptase. If that proves to be true, it's another way in which this virus, which at first we thought couldn't possibly remain latent in the body, might be able to do so.
Interesting. We have a couple questions in the chat box. How does the data you have presented connect to dysautonomia, specifically POTS, which we so often see in post-COVID?
Yeah, well, there's no doubt that dysautonomia is well documented in both long COVID and ME/CFS, but I think we really don't know why. What triggers it? Is it some central effect of the virus which can infect the brain on the brain stem where the autonomic function is coordinated? That's where I'd put my money, but I don't know enough about autonomic physiology to make a very educated guess. I think that's the most likely explanation. But there's no doubt at all that dysautonomia can be quite prominent in both long COVID and ME/CFS, can produce many of the symptoms of that are common in both of those syndromes. By altering vascular flow in the brain, for example, with ME/CFS, there's evidence that with an orthostatic challenge, blood flow in the brain drops 30%, whereas in healthy control subjects, it drops, but about 2%. So there are many ways in which dysautonomia could lead to some of the symptoms of long COVID and ME/CFS. But why the dysautonomia? I think that's more speculative.
Thank you. How do testosterone levels or other sex hormone levels affect immune function and COVID recovery?
I don't know the answer to that. In fact, the only fairly robust hormonal evidence I'm aware of is, again, both in long COVID and in ME/CFS are low cortisol levels. Not Addisonian range, but very low normal or just below normal. In blood, in urine, at the right time of day. So it's That hormonal change has been well established, but there may be sex hormonal changes, not just the sex hormones, but the gonadotropin releasing hormones, abnormalities in both of these conditions, but I'm not aware of evidence for that.
Jen Bell has a question.
Hi, I was curious just to get your impression of the value of doing herpetic testing or testing for herpes viruses, not only to evaluate presence of past infection, but for reactivation, and how valuable that testing is, and if it can be used to help guide treatment, particularly like the EBV testing, EBV panels. And I mean, I run them on these long COVID patients, but sometimes I'm not sure what to do with the information.
Right. Yeah, I'm afraid I agree with you. I'm unsure for two reasons. One is that in my experience, seeing subtle abnormalities that suggest reactivation, for example, of Epstein-Barr virus, like finding antibodies to the early antigens of the virus. That was thought to be pathognomonic of significant reactivation of the virus. But 30 years ago, expecting that's what we would find, we and others found that no, actually there are a lot of healthy people walking around with low levels of these so-called pathological antibodies that signal reactivation. When you see really striking elevations in the IgG to the viral capsid antigen of EBV, or striking elevations of antibodies to early antigens, I think take that seriously. That almost surely means there's a lot of viral reactivation going on. But then you come to the second obstacle, which is, has it ever been shown that using an antiviral, which should work if in an actively replicating virus, actually improves the situation? And I'm not aware of any studies that have shown that. So I'm not real aggressive about ordering herpes virus antibodies. I've been waiting for a test, which is more likely an antigen or nucleic acid test that's very sensitive, that will discriminate cases from controls just as dramatically as that data I just showed on SARS-CoV-2 spike protein, where 60% of the long COVID cases and 0% of the healthy controls had the signal. Now, that's something, if it proved to be true, that kind of test for a virus would really be useful information, but I don't think we have it yet with the herpes viruses.
Thank you.
I think one of the things that I've been lucky enough to be involved in is we are part of the trial looking at nintedanib for these post COVID fibrotic patients, because like sometimes, you know, when you talked about like all the potential mechanisms, it'd be interesting to see the overlap, because some of our patients that started out with an autoimmune disorder, and it's sort of like kind of like the old 1-2 hit hypothesis. It's like you start out with an immunological tilt, then you get an insult, then you get this. So I'm curious, they will actually have an info on some of these patients, like autoimmune antibodies and some of these things, and looking at how they respond, because I kind of wonder if you're just seeing, you know, sort of some people are predisposed to have some of these issues. You know, it just happens to be this particular insult versus an herpes virus or something else. So to be seen.
You know, Dr. Brown, I agree with you. In fact, there's one paper, I can't remember the citation off the top, which found that a predisposition to produce autoantibodies that predated acute COVID predicted autoantibodies and long COVID, suggest very much consistent with your hypothesis that some people are genetically vulnerable to the autoimmune consequences of acute SARS-CoV-2 infection. And that may be a risk factor if it could be more systematically documented.
It'll also be interesting to see if the gender tilt of these predisposition also kind of correlates as well, because we're seeing a lot more women than
Yeah.
men, and other factors. So
Yeah, it's really interesting that, as I understand the literature, the severity of acute COVID is greater in men, but the likelihood of long COVID developing is greater in women. And that's also true of ME/CFS, and it's also true of most autoimmune diseases. And it's also true of autoimmune disease models in animals, where the females of the species are much more likely to develop the autoimmune disease. And when you give the males of the species who are asymptomatic female hormones, they then develop the autoimmune disease. So I really do think it's likely that there is a sex hormone involvement in many autoimmune diseases. Whether that's true in this disease, I don't think is established yet.
Oh, interesting. We did have a question from earlier, so this is kind of open to the mentor panel in general. Do you recommend anti-inflammatory diets in patients? And I'm assuming that's with long COVID and likely ME/CFS.
Yeah. Well, I don't think there's a lot of evidence that the conventional anti-inflammatory drugs, by which I mean NSAIDs and steroids, do much, or the potential benefit is worth the potential adverse effect, particularly with steroids. The one exception to that may be the growing literature on the use of low-dose naltrexone in ME/CFS and in a number of other diseases characterized by inflammation. There's some animal evidence that low-dose naltrexone quells neuroinflammation. And you could argue, if you believe the model I presented today, that neuroinflammation is at the center of what's generating the symptoms, then it would make sense to try to use a quite benign treatment to quell neuroinflammation and see if it improved the symptoms. I hope that that's one of the treatments that the NIH RECOVER study tries. The only one that I'm aware they've committed to funding a trial of is Paxlovid. But I would, top of my list would, from what I hear from colleagues who have used low-dose naltrexone, is that drug. I wonder if Dr. Bateman and Dr. Yellman might comment on that.
I'm happy to comment on it. We use a lot of low-dose naltrexone, and I completely agree with what you described. Tony, I want to ask a harder question, which is, do you think anti-inflammatory diets are helpful? And anybody else who wants to answer?
Well, I have no experience with that in long COVID. So with ME/CFS, I will say this. I'm impressed by the patients that I've seen over the years who swear by a certain diet. I wouldn't label that as anti-inflammatory diet until I had robust markers of inflammation that correlated with using that diet. But there are people who absolutely are convinced that certain diets improve their symptoms and that other diets make their symptoms worse. Now, whether they're doing that via generating inflammation or some entirely different mechanism, I don't know. That's all I could say.
I'll add to that, and I don't know either, but I usually encourage people to, eat less sugar and highly prepared foods, and keep less chemicals out of their diet as much as they can, because what I've seen is the common denominator on diets that people swear by, right, are more natural foods, lower carb, lower sugar, Mediterranean diets, and then, there are diets like the low FODMAP diet, right, that have been established to help in certain inflammatory diseases. And so there's no reason not to try some of those things. And if you think about mast cell activation, there may be certain foods that trigger mast cell responses more than others. And if you can identify what those are, that might be a start, right? Because the approach to allergies is always avoidance first, right?
Yeah.
And then to try to blunt the immune response and get the process that's revving it up ramped back down. So whatever doesn't people, as long as it makes things better and doesn't make things worse for people.
Great.
That would be my guidance on diet.
Are there any additional thoughts on that?
I agree. The only other thing too, I've seen people that have done a lot of like extreme diets or other things, nutritional deficiencies can creep in or other issue. The other thing I see is like super supplementation, people taking massive doses of vitamins, and I always advise people on like fat soluble vitamins are not eliminated. So we have seen people with like super crazy high vitamin D levels and things. So it just, caution people from doing, those sort of things.
And I would just point out to what Dr. Laura Pace mentioned at a previous echo is that to our knowledge at this point, trying to modulate the gut microbiome to affect this type of inflammation, we don't have the strategies quite yet. We're not there yet.
Okay, I think we have time for one more question in the chat here. Is there evidence that COVID vaccines reduce the chance of developing long COVID?
Yeah, I think there's now a lot of evidence. I just saw numbers earlier today from a large study that something like 87% of the people with long COVID in a large study were unvaccinated. And several studies of the impact of groups, prospective studies comparing people who got vaccinated to those who didn't get vaccinated when they all got COVID, found that there were fewer long COVID cases among the vaccinees than the non-vaccinees. So yeah, I think that's just another one of the benefits of vaccination, along with not dying.
I think, I can't remember the reference right now, but there was a study that came out of Italy, really big study, that showed that the percentage of people who went on to develop long COVID went down with each variant and with each every stage of vaccination. So I think that's good. I think I'd like to see some more studies of that size, but it was pretty impressive the way they collected their data.
Yes, that's true, especially Omicron compared with Delta. There was the big difference.
I think it's hard to sort out the influence of vaccines versus variants. I think it's impossible to, but at least we know that as we've gone down the path, the percent of people who get the infection, who go on to have long COVID, is going down. The problem then is the number of cases are still very high.
Exactly.
So it doesn't take a very high percentage of people who get COVID to have a large number of people with long COVID, even if it's a very small percent. And I would say, and Tony can confirm this, it depends on the study, but long-term in other post-viral syndromes, the thinking is that it's probably, I don't know, around 0.5 to 1% of people go on to be sick indefinitely after their infection. A lot of them have gone out to 3 years, but EBV studies, prospective EBV studies, and about 3% of those people at 3 years, and in that study they were all women, because by the time you get out that far in a prospective study, the number of cases is really low.
Yeah, the problem with most of the studies of post-infectious fatigue syndromes is because it's hard and costly. Very few studies have followed people for a very long time. There's lots of studies have followed people for a year, 18 months, but then it drops off. And so I think we honestly don't know about the natural history of most post-infectious fatigue syndromes.
I feel like COVID is kind of the gift that keeps giving regardless of what we want it to. But in some ways, I share the optimism that you expressed that we'll learn a lot about things that we never really got to know about well, like fatigue, post-exertional malaise, and hopefully also do the engagement of patients with, I feel like COVID has been very different in comparison to other diseases. Patients are very altruistic and want to be part of studies. So, and patient-centered outcomes, I think, has been more focused on for this research, I feel like, as well. So very cool. Thank you so much for your presentation.
That was
It's a
great.
pleasure. Thanks for having me.
A lot
Bye-bye.
of wonderful discussion. Thank you, Dr. Komarov.
My pleasure.
Keep up the good work.
So just to wrap, thank you all for joining today. Our next session will be on November 15th. We'll be hearing from Dr. Yelman on the topic of headaches. He's going to kind of tie all this together for us. And then our last session of 2022 will be on December 6th, and we'll be hearing from a specialist from Barcelona, Spain, who's going to be speaking on hypermobility, EDS, CCI, tethered cord, and AAI. So hopefully we will see you guys then. Take care and stay safe.